I want you to imagine your brain as this incredibly complex, bustling city switchboard. You have billions of lines connecting, flashing, transmitting messages back and forth every single second of your life.
Right. It's just constant, nonstop activity.
Exactly. But what happens when a specific set of those lines gets completely overloaded? Say a massive surge of activity just hits the system. They're ringing constantly, creating this absolute electrical chaos. How does your brain restore order?
Well, it turns out it literally cuts the cord.
Yeah, which is just wild.
It is. It's a ruthless but entirely necessary kind of biological triage. If a circuit is too noisy, you just dismantle it. And for a long time in neuroscience, I mean, we understood that this pruning happened.
Right, the brain cleans up after itself.
Exactly. But the exact mechanism, like the literal microscopic shears doing the cutting in an adult brain, was a bit of a black box.
Which is exactly our mission for today's deep dive.
Wow.
We're unpacking a truly groundbreaking paper published in Science by Crowley and colleagues. And the reason this paper is making such massive waves is that, well, they didn't just find the shears.
No, they found out who is holding them.
Right. We're talking about a completely unexpected, almost unbelievable collaboration between your brain cells and your adaptive immune system.
And that's the part that is really sending shockwaves through the field right now. I mean, the idea that immune cells, the very same ones we normally associate with, fighting off a cold or recovering from a bacterial infection, are actively participating in shaping our neural circuits.
Our thoughts and our memories. It's genuinely amazing, like antibodies in the brain calling the shots.
Yeah, it rewrites a lot of textbooks.
It really does. So today, for you listening, we're going to explore exactly how this pruning mechanism operates in a healthy brain. But more importantly, we're going to look at how this exact same system goes completely off the rails in early Alzheimer's disease.
And crucially, how researchers might actually be able to hit the brakes on that destructive process one day.
Yeah, that's the holy grail, right? So let's start at the beginning. If we have a noisy, overloaded brain circuit, what is the trigger that actually starts the destruction?
Well, to figure that out, the research team had to create that overloaded circuit artificially in a lab setting.
Because you can't just wait around for a mouse to get stressed out.
Exactly. So they started with healthy adult wild-type mice, and they zeroed in on a very specific neural pathway, the connection from the entorhinal cortex to the hippocampus.
Okay, let me pause you there. For those of us who aren't neuroanatomists, why that specific pathway?
That's a great question. The entorhinal hippocampal circuit is essentially, well, a classic memory highway. It's crucial for learning and memory formation.
Right.
But a neural circuit is just a series of connections between neurons. You can, picture a neuron like a long wire, and at the end of that wire is a terminal.
Like the plug that connects to the next cell.
Precisely. It's the synapse, the gap where the message jumps across.
Okay, so they have their target memory highway. But the challenge is, how do you force just one specific wire in this incredibly dense brain to become hyperactive?
Right, without accidentally causing a massive seizure or, messing up the rest of the brain's activity.
Yeah, you can't exactly just plug a tiny battery into a single cell.
You really can't. So they used an incredibly elegant technique called chemogenetics, specifically a system known as DREADheads.
DREADs. Okay, I saw this acronym in the paper. It stands for designer receptors exclusively activated by designer drugs, which is a massive mouthful.
It is a bit of a tongue twister.
Explain this like I'm 5. What are we actually doing to the mouse here?
Let's break it down. Imagine you could install a custom remote-controlled volume knob on a very specific set of neurons.
Okay.
To do this, researchers use a harmless viral vector. They inject it into the entorhinal cortex. And this virus essentially delivers a genetic instruction manual to just those specific neurons.
And what did the instructions say?
They tell the neurons to build a synthetic receptor called an HM3D Hue receptor and put it on their surface.
Okay, so now these specific memory neurons have this new artificial receptor on the outside, but it doesn't do anything yet, right?
Exactly. It does absolutely nothing. It just sits there, completely ignoring the brain's natural chemicals.
Got it.
But when the researchers inject the mice with a specific synthetic drug called CNO, that drug acts like a custom key. It perfectly fits the new designer receptor.
Oh, wow.
Yeah, and when it binds, it turns those specific neurons on. It forces that exact circuit into a state of intense hyperactivity.
That is just wild. You essentially build a custom lock on a specific brain cell, and then you just hand the mouse the key to turn the cell on.
That's a perfect way to put it.
So they cranked the volume to 11 on this memory highway. What actually happened to the switchboard?
The hyperactivity directly caused the loss of very specific presynaptic connections at the receiving end of the circuit in the hippocampus.
Okay.
Specifically, they saw a significant drop in what are called VGLUT2 terminals.
Now wait, why is it important that they were VGLUT2 terminals? Because I remember the paper making a big deal out of the fact that neighboring terminals, like the VGLUT1 terminals, were completely fine.
Oh, it's a crucial detail. VGLUT1 and VGLUT2 are basically just, different types of plugs at the end of the neuron wires.
Right.
The fact that only VGLUT2 was destroyed means this wasn't a carpet bombing situation. The brain didn't just panic and shut down the whole neighborhood because of the noise.
It selectively, surgically pruned the exact specific connections that were overwhelmed by the hyperactivity.
Exactly. Surgical pruning.
Okay, so we know the noise, the hyperactivity, causes the loss. But the noise itself isn't like a physical pair of scissors. What is the physical tool doing the actual cutting?
That brings us to a protein called C1q.
C1q, I feel like I need a scorecard for all these terms.
I know, the alphabet soup gets a bit dense here. But C1q is fascinating. It's the initiator of what immunologists call the classical complement cascade.
Okay, you're going to have to translate classical complement cascade for me.
Sure. Think of the complement system as a domino effect within your innate immune system.
The innate immune system being your body's first line of defense, like the general cleanup crew.
Right. So when C1q binds to a surface, it knocks over the first domino. That triggers a cascade of other proteins that eventually assemble into a complex that either, eats the target cell or punches a hole in it.
Usually we think of that clearing out dead cells or bacteria in the bloodstream, right?
Yes, but in the brain, C1q acts as a kind of molecular trash tag.
Okay, I love that visual. So let me try an analogy here. It's like C1q is an arborist's bright orange tape.
Oh, I like that.
You have this overgrown, hyperactive VGLUT2 branch on a tree, and C1q comes along and wraps an orange tape around it. And that tape signals the immune system's cleanup crew, the rest of those falling dominoes, to say, hey, chop the specific branch down.
Yes.
But the neighboring VGLUT1 branch has no tape on it, so the cleanup crew just walks right past it.
That is a perfect way to visualize it. And it's exactly what they saw under the super resolution microscopes.
What?
The hyperactive mice had a massive increase in C1q deposited specifically on those overworked VGLUT2 terminals.
But wait, how do they know the orange tape is what actually caused the branch to get cut?
Well, here is how they proved it was the cause. They repeated the entire experiment in mutant mice that were genetically engineered to lack the C1q protein entirely.
So they just took away the orange tape.
Exactly. They turned on the designer drug, the circuit went completely hyperactive, but...
Let me guess, no synapses were lost.
No synapses were lost. Zero.
Wow. No tape, no cutting. Okay, so if you're listening and trying to keep track, let's do a quick recap. We've basically proven two things so far.
Go for it.
First, just the sheer electrical noise of a hyperactive neuron is enough to trigger its own destruction in an adult brain. And second, this C1q protein is the physical orange tape that marks it for death. Am I missing anything?
You've got it perfectly. But naturally, this mechanism raises a massive question.
Right. What happens when things break?
Yeah, if causing hyperactivity destroys synapses in a healthy brain, what happens in a diseased brain where hyperactivity isn't just a, temporary drug effect, but a chronic, ongoing problem?
Right. And this is where the paper shifts from basic biology into a potential Alzheimer's rescue mission.
Yeah.
Because this pruning system doesn't just do healthy maintenance. Under the right conditions, it goes totally rogue.
It really does. And to study how it goes rogue, the researchers use something called the J20 mouse model.
Let me guess, more genetics.
Always.
Oh.
The J20 model is genetically engineered to mimic early-stage Alzheimer's disease.
Okay.
Now, when most people think of Alzheimer's, they think of amyloid plaques, those physical clumps of protein that build up in the brain.
Yeah, that's what always makes the headlines.
Right. But a key, often overlooked feature of early Alzheimer's is rampant neuronal hyperactivity. This electrical chaos happens long before you see the physical plaques forming.
Oh, wow. So the switchboard is sparking and short-circuiting before the physical wires even look damaged. Okay, so in the first experiment, they installed a custom volume knob and turned the brain's volume up. To see if they could rescue the Alzheimer's brain, they need to do the opposite, right?
That's it. They used a different type of dread this time, an inhibitory receptor called HM4DI.
Okay.
So when they administered the designer drug, instead of firing up the circuit, it forced the naturally hyperactive Alzheimer's neurons to quiet down.
They chemically forced the brain to just take a deep breath and chill out. So what happened?
The results were remarkable. By artificially dampening the hyperactivity, they saw a massive reduction in the local production of amyloid beta, the toxic protein that forms plaques.
Just from turning the volume down.
Just from that. They also saw a significant drop in the deposition of our orange tape, that C1q tag.
Amazing.
And most importantly, they saw that the postsynaptic terminals, which they marked by looking at a protein called Homer1, were actually partially protected from destruction.
Wait, I want to clarify something for the listener. Earlier, we talked about presynaptic terminals getting destroyed. Now we're talking about saving postsynaptic terminals.
Good catch, yeah. A synapse has two sides. The presynaptic terminal is the side sending the message. The postsynaptic terminal, where we find Homer1, is the receiving dish on the next neuron.
Right, so if you destroy the sender, the receiver usually degrades too.
Exactly. By turning down the noise, they saved the structural integrity of the receiving end.
Okay, here is where my brain gets stuck, though. How does turning down an electrical signal stop the physical buildup of toxic amyloid beta and stop the immune system from laying down C1q? I mean, how does electricity change biology like that?
It all comes down to breaking a vicious cycle. The working hypothesis in the field right now is this brutal self-feeding feedback loop.
Okay.
It goes like this. Hyperactivity in the neurons physically causes the cells to release more amyloid beta into the gap. That amyloid beta then aggregates and irritates the neurons, which causes them to become more hyperactive.
Oh man, so it just feeds itself.
It completely feeds itself. And this runaway electrical noise acts like a distress beacon. It summons C1q, which starts mercilessly tagging synapses for destruction in a desperate attempt to quiet the system down.
By just destroying the connections entirely.
Exactly.
Okay, so it's exactly like a screeching audio feedback loop at a live concert.
Oh, I love that.
Yeah, think about it. The amyloid beta is the microphone, and someone has placed it way too close to the speaker, which is the electrical hyperactivity. The noise goes into the mic, comes out the speaker louder, goes back into the mic. It just loops and gets deafening.
Right.
And C1q is the panicked sound guy running over and just ripping the audio cables out of the wall to save the speakers.
That is a phenomenal analogy. Yes, that is exactly it.
But by chemically turning down the volume of the brain activity using those adreds, the researchers basically just grabbed the microphone and pulled it away from the speaker. They broke the loop.
They broke the loop. You stop the hyperactivity. You stop the excessive amyloid beta release. And because there's no distress beacon, the C1q sound guy stands down and the actual synaptic cables are spared.
That is a massive, massive insight for potential future Alzheimer's therapies.
It really is.
So just to lock this in for everyone listening before we get to the real plot twist, early Alzheimer's brains get caught in this screening feedback loop where noise creates toxic proteins, which creates more noise, ending in synapse death.
Right.
But by artificially silencing the neurons, we basically unplug the mic and save the synapses from the immune system.
Exactly right. But we're still missing a crucial piece of the puzzle here.
Which is?
We know C1q is the orange tape, the pruning shear. We know hyperactivity triggers the process. But C1q is just a protein floating around in the tissue. It's a dumb tool.
Right.
It needs a very, very specific signal to know exactly which synapse to tag. Remember, it only tagged the hyperactive VGLUT2 terminals, but spared the VGLUT ones right next door.
Oh.
Something has to be pointing the finger. Where is that targeting signal coming from?
Right. Something has to tell the sound guy which specific cable to pull. And the answer to this is what completely blew my mind when I was reading the sources.
It truly shocked the researchers, too. I mean, to find out what was directing C1q, they used advanced spatial transcriptomics. Specifically, they used commercial platforms called Visium and Xenium.
Spatial transcriptomics. That sounds incredibly dense. What are we actually looking at with that?
Think of it like taking a microscopic slice of brain tissue, and instead of just taking a photograph, you're mapping exactly which genetic blueprints, the RNA transcripts, are actively being read and exactly where they are located down to the single cell level.
Wow, okay.
Yeah, imagine looking at an entire city grid and being able to see exactly which factories are printing which instruction manuals at any given second.
Okay, so they're looking for the factory that's printing the instructions to guide C1q, and they were assuming they'd find microglia, right? Because microglia are the brain's resident immune cells. They're the local housekeepers.
That was the absolute expectation. But instead, the transcriptomic map lit up with transcripts for immunoglobulins, specifically IgG, IgA, and IgM.
Which are antibodies.
Yes. And they found the specific B-lineage cells, the immune cells, that actually manufacture and secrete those antibodies.
Okay, hold on. Stop right there. I have to push back here because everything I learned in basic biology says this shouldn't happen.
Right.
Antibodies in a healthy brain. What about the blood-brain barrier? I mean, the brain is supposed to be an immunologically privileged site. It's a walled garden that explicitly keeps the adaptive immune system like B cells and antibodies locked out so they don't cause massive inflammation. Are you saying the textbooks are wrong?
It is a total paradigm shift.
Yeah.
You're completely right. The brain tissue itself is heavily guarded. So when they looked closely at the spatial map, these B cells weren't floating deep inside the brain tissue itself.
Where were they?
They were parked in what are called the leptomeningeal niches. The leptomeninges. They're the membranes that wrap around and form the protective borders of the brain. The B cells were sitting in these border walls, situated directly adjacent to the hyperactive hippocampus.
Just sitting at the border wall looking over the fence.
Exactly.
But where did they come from? Did they cross over from the general bloodstream?
That's exactly what they wanted to know. To track their origin, they used an incredibly clever tool called Cade mice.
Cade mice. What makes them special?
Well, these mice are genetically engineered so that all the cells in their body naturally glow green under a microscope.
Okay, green glowing mice. Got it.
Right, but here's the trick. If you shine a very specific wavelength of UV light onto a tissue, the cells in that spot permanently switch from glowing green to glowing red.
Oh, wow. So it's like a biological highlighter.
Yeah.
You just shine a light and highlight a specific group of cells in red so you can track exactly where they go later.
That is exactly what it is, a biological tracking device. So the researchers suspected the skull might be involved. They shined the UV light solely on the bone marrow of the mice's skulls, turning those specific marrow cells red.
Okay, and then?
And then they triggered the brain hyperactivity using the designer drug. When they looked again, they found those red glowing B cells had actually migrated out of the skull's bone marrow, traveled through the dura membrane, and parked themselves right at the border of the brain.
Right next to the hyperactivity.
Yes.
Let me just process this because it sounds like science fiction. The brain gets overwhelmed by electrical noise. It somehow sends an SOS signal straight through the bone to the skull marrow.
Yep.
The adaptive immune system hears the call, marches B cells out of the skull bone, parks them at the brain's border, and they start manufacturing and shooting antibodies into the brain tissue itself.
That is exactly what is happening.
That is wild. Our walled garden isn't just letting border guards in, it's actively requesting them from the skull.
It's a fantastic discovery, but, as any good scientist will tell you, correlation is not causation.
Right. It's one thing to find B cells and antibodies at the scene of the crime.
Exactly. It's another thing entirely to prove beyond a shadow of a doubt that those specific antibodies are the ones pulling the trigger and guiding the C1q pruning shears.
Because how do we know they aren't just there to clean up the mess afterward? How did they actually catch them in the act?
Well, they started by looking closer than ever before. Using super-resolution air-skin microscopes, they could physically see individual molecules interacting.
And what did they see?
They caught it red-handed. IgM antibodies were physically touching and co-localizing directly with the C1q tags right on those specific overworked VGLUT2 synapses.
So the antibody's physically touching the orange tape on the synapse. It's at the scene.
Yeah.
But to prove it's strictly necessary for the cutting to happen, you have to break the system again, right? You have to remove the suspect and see if the crime still happens.
Exactly. And they did this through 3 brilliant sequential knockout tests.
Let's hear them.
First, they use what are called MoMT mice. These mice are genetically altered, so they completely lack B cells entirely. No B cells, which means absolutely no antibodies.
Okay.
When they forced the brain circuit to become hyperactive in these B cell deficient mice, there was 0 C1q deposition and 0 synapse loss.
Okay, so if you remove the B cells, the pruning stops. But let me play devil's advocate here.
Go for it.
Maybe the B cells are doing something else, like maybe they secrete a different chemical, or maybe just their physical presence is required.
Yeah.
How do we know it is the antibodies doing the work?
That leads directly to the second test. They used CyGM KO mice. Now, these mice can make B cells, and those B cells can make antibodies, but they possess a tiny mutation that prevents them from secreting the IgM antibodies.
Oh, I see.
Yeah, the antibody just stays stuck to the surface of the B cell. It can't float free into the tissue.
So the B cell is there, but its primary weapon is jammed in the holster.
Exactly. They force the hyperactivity again, and again, no synapse loss.
Wow. So it definitively has to be secreted IgM floating into the brain. But wait, antibodies are famous for being highly, highly specific, right? Like a lock and key.
Yeah.
your COVID antibody doesn't fight a flu virus.
Right.
So is this IgM recognizing a very specific stress signal on that overworked synapse? Or is it just acting like sticky glue?
That brings us to the most elegant test of all. The third test used MD4 mice.
MD4 mice.
Yes. These mice have healthy B cells, and they freely secrete IgM. But their genetics are hijacked so that all of their IgM is programmed to target one specific fake antigen hen-egg lysozyme.
Hen-egg lysozyme. Wait, a protein from chicken eggs.
Yes. A protein that would obviously never, ever exist naturally in a mouse brain.
Ah, so the mice are secreting tons of IgM, but it's completely blind to anything in the brain because it's frantically searching for chicken egg protein.
Precisely. The lock is there, but there are absolutely no keys in the brain that fit it. They triggered the hyperactivity in these mice, and once again, 0 C1q deposition, 0 synapse loss.
Oh, that is definitive. That is a mic drop right there.
It really is.
Okay, I want to try a new analogy to cement this whole complex mechanism together for the listener, because we've moved way past the orange tape at this point.
Let's hear it.
Let's say the secreted IgM antibody is a highly calibrated laser pointer, and the C1q protein is a guided missile.
Okay, I like where this is going.
When the brain gets overloaded, it calls the B cells to the border wall. Those B cells start shining this highly specific IgM laser pointer through the brain tissue, and only illuminates the exact overworked VGLUT2 synapse that is flying off the handle.
Yes.
Without that precise laser painting the target, which is the antigen-specific lock and key action of the IgM, the C1q missile stays home. The cleanup crew never gets the signal, and the synapse survives no matter how chaotic and noisy it gets.
That is a phenomenal synthesis. It proves definitively that secreted antigen-specific IgM is an absolute requirement. It is the literal physical bridge between the adaptive immune system, the B cells, and the IgM, and the innate immune system, the C1q.
They have to physically link up to execute the pruning.
Exactly.
So if I'm taking a step back here, the implications of this are staggering. And why should you, listening to this right now, care about mouse brain biology?
Because it applies to all of us.
Right. Because every single one of us experiences stress, intense bouts of learning, sleep deprivation, and just the natural process of aging. All of those involve your brain circuitry getting noisy and needing remodeling.
Absolutely.
And knowing that your highly individualized adaptive immune system is actively making the structural decisions about your brain circuitry changes everything. Because my immune system isn't the same as yours.
It's not. Your immune system is as unique as your fingerprint. Your B cells carry the immunological memory of literally every single pathogen, every flu, every scrape you've ever encountered in your entire life.
Wow.
And this raises an incredibly provocative question that the paper points toward, but, doesn't strictly answer.
Hit me. What's the question?
If our adaptive immune system learns and builds its antibody repertoire based on a lifetime of fighting specific environmental viruses and bacteria, could the completely unrelated infections you fought decades ago uniquely influence which memories or which cognitive functions your brain decides to prune as you age?
That is mind-bending. Are you saying the bad case of the flu I had in my 20s could theoretically dictate which synapses I lose in my 70s?
It's possible, because if the B cells are the ones deciding what the laser pointer looks for, their past experiences have to matter.
That is incredible.
It strongly suggests that our cognitive trajectory, how our brains age and maintain function, isn't just about what happens strictly inside the brain. It is a historical record of our immune system's entire life journey.
So the next time you feel like your brain is an overloaded city switchboard, just remember, before a single cord gets cut, a whole team of immune cells from your skull has to sign off on the work order, and they are bringing your entire medical history with them. Thank you for taking this deep dive with us today. Keep questioning the connections.